Hello, welcome to Dees Talk. Today, my guest is Timi Ellinas. She is the CEO and founder of Helene Therapeutics, which is the first GMP-certified autologous stem cell center in the Western world. She is a researcher, biotech leader, scientist, entrepreneur, and pioneer.
Ooh.
Let's get into it.
Wow, what an introduction. Actually, I was always like that.
Yeah?
Never really self-conscious, no. I like to also do like little, I dress in themes. I think when I want to feel glam, I do glam. When I want to, you know what I mean? Like I'll do theme, like maybe I want to.
Dress for the occasion.
Kind of thing. Even if it's not the occasion though, I think.
Right. Oh, really? Like you're like.
I'm like, I want to overdress today. Yeah, but you'll be very overdressed. Yeah, fuck it.
Yeah, it's better to be overdressed than underdressed, right?
I can be underdressed as well. Like if I want to be, I don't know, like a skater chick, I'll do that. Right. If I want to be 80s punk, I'll do that. Oh. If I want to be glamorous opera singer, I'll do that.
Right.
I'm just like, and my repertoire is.
Are these part of your like previous identities that you kind of like moved on from, but then keep in the back pocket in case you need to pull it out?
Probably.
Yeah,
probably.
I find that as well. I've got a few of those personas.
The thing is that I'm black and white at the same time, but never gray.
Right. Black and white at the same time. In what way?
But never gray. Huh?
Like grays. You're like extreme. No.
Yeah, extremities. Yeah.
Yeah, that's good.
Like I love my favorite band is Tool.
Tool.
Do you know Tool?
No. What are they?
Well,
are
they
punk
invented
rock?
new metal.
Oh, okay. So is that quite like headbanging stuff or what?
Yeah, a bit more melodic, but yeah, it's you know, like intense drums. Voice is softer. Maynard's voice is beautiful.
So like mosh pits and all that. Or?
I don't know.
Do you like to
possibly.
mosh pit? Get down in the mosh pit now?
I've done that as well.
Yeah.
I love opera. I love classical music. I mean, I was born in LA, so rap.
Yeah, sure.
You know what
I mean, I always think of, this should be on the podcast. Are you rolling, Jeff, or what? I always think of like, you know, the samurais that they're like these warriors that they go out and they have to learn all these different kind of fight techniques and sword techniques and grappling. But then they also learn about like poetry and like culture and all that. And they take that just as seriously. And it's like, you know, you want to be as a kind of rounded human being. You don't want to have just like, oh, I am this and that's it. It's like, there's so much going on. And I think.
And why can't we like everything? Yeah. You know what I'm really pissed off with the world, where we've come to today. Everyone, you have to be a specialist and specialist and specialist. So much so that we're just in this little tiny box. Yeah. And at least in our field, that doesn't work. Because the body is one system.
Absolutely.
You need to be able to connect the dots. And if you don't know what box is over there, and then how are you going to connect what's happening there?
It doesn't even make sense they don't teach it that way, because it's not like you can't just isolate a liver. Yeah, because everything flows into one another. It's like one whole system. It's like learning about a car, but just learning about one part of the car and just specializing in that. And then you kind of fix that bit and pass the problem onto some other part, which seems to happen quite a lot when they're like, we'll take a drug to fix this one problem, but they're not looking at it holistically and kind of healing the whole body.
And it's as if they don't believe you as well. I always remember this example that my father had given me. He had, once upon a time, an interview. And in Greece, we really do focus in on lateral thinking. And the person that was interviewing him, I think she was either English or American, but I think, anyways, she was from a northern country. And she pointed out to me, she said, well, you know this, and you say you also know this. How can you know both of these things? Like kind of trying to call him out that he was lying about his skill set. Right. And he was just totally confused. And she was totally confused because she couldn't fathom that he was an expert in two different areas.
More than one thing.
In more than one thing, which is crazy. And it's also like, why are we like underestimating ourselves as humans? Don't people always say that, well, the brain, it's a marvel,
It's
and we only use
capable.
10%.
Yeah.
And then it's like, but they don't let you use
Yeah,
your whole
also
brain,
there's
and
so
they don't
mu-
believe that you can use your whole
Also,
brain.
there's so much stuff you will learn in one field, which you can kind of bring into other fields. And the more different fields you have little insights in,
Yes.
you can be like, if everyone's just too narrow, they don't understand that, that technique or whatever it is from one thing you could bring into something else. And then, get breakthroughs as well.
Well, exactly. So in my field, I remember one of my lecturers, Professor Brian Sutton. Actually, we saw him recently at our stem cell center, but about 20 years ago, I remember something that he had said about solving the structure of a specific protein, because way back when, not anymore really, but way back when, it took approximately 10 to 20 years to solve a structure of a protein.
Is that the protein folding that they're talking about? Or is that what?
Well, yeah, the structure of it. Not
Sure.
just how it folds, but the whole structure. So proteins are a string of amino acids,
Right.
and the way that they fold together, their tertiary structure, makes it a protein.
Right.
And the one thing that they were saying about a specific protein, and excuse me, I don't remember which one precisely, 20 years ago now, was that someone from a different field they would expect would come in and solve that structure.
Sure.
Just exactly what you're saying, just an open perspective.
Yeah. There's a book that I'm actually just reading at the moment that's all about this. It's called Range. Have you heard of that? It's like really worth reading. I can't remember who's the author name, but yeah, they speak about exactly that, you should have, it's a bit of a kind of myth that just specializing in one thing makes you excellent at that. Or, I mean, it can be for certain types of skill sets. Like if you're a golfer and you just repeat the same thing over and over, then you kind of repetition in a skill set can get you closer and closer to be more accurate or whatever it is. But if you're, most
muscle memory, that kind of thing. However, Arnold Schwarzenegger, which was my first true love.
Oh, really?
Yeah, he was. He really was. I was in love with him.
Oh my God. I can see why.
My father did a lot of bodybuilding when he was younger. So back in LA, so I was born in LA. Everything that we do right now here is what we did.
What? Move it.
40 years ago.
So what? So Arnold was your crush? When you were young? Why?
Yeah, big time. It really was. So that, as I was saying, my dad has a particular interest in bodybuilding. So, I mean, he knows and still keeps in touch with some of the Mr. Olympias from back then.
Oh, so he's down with them.
Oh, he's down with them,
right.
Yeah,
and does he work out himself a lot?
Yes,
then?
we have a gym on the top floor of our house in Greece is a gym. Right. He's got 30 instruments.
Right. What, like
workout.
A workout stations or whatever. Right.
Yeah.
Okay.
So he works out twice a day.
How old is he now?
He's 69.
Wow.
In February, he's going to be 70.
And he's still working out twice a day.
He is. So we took a sample of his stem cells.
Yeah.
And he's got the best stem cells.
Really?
But he's been doing this longevity stuff since I was born, really. So funny story. We used to go to Muscle Beach where he would work out and, in Venice, and we'd stop by one of these health food stores, and we used to have green juice shots, they were wheatgrass.
Sure.
Which, I mean, quite interesting for a 3-year-old to...
Oh, really? Yeah, it's quite harsh.
He started me young.
Yeah, it's quite harsh, yeah.
It is. But I loved it. My breakfast was a protein shake with raw egg.
Oh my God.
Yeah. He would make
it. I mean, can you give that to kids? Because I make protein shakes in the morning as well. And it says on the packet, not for under 16 or something, I think. Well, you turned out okay.
I turned out okay. And I actually started training with this incredible PT, Kyle, which we've started working with as well, because we want to do a whole 360 patient journey. And it's really important because, as we were saying, your body is one system. It really gets to me when people say body and mind, but your mind is part of your body.
Of your body, yeah. Your brain, yeah.
Body. Yeah. That's one system.
Sure, yeah.
F of X, that's just one whole system. And we need to treat it like one system. And we measured my the percentage of fat and muscle. And I haven't worked out for a very long time, unfortunately. I just started now again with him because I've just been too busy. Like, there's no time and 2 little kids. I mean, you know, I would love to.
How old are your kids?
Constantine is 5 and Leander is 9.
Okay, right.
Yeah.
So.
And they also love working out. They're all about Mr. Strong.
Oh, really? Both of them?
Well,
That's
yeah,
good.
yeah.
Well, it's
They
good
look
to get
up
the
to my
kids
father.
into exercise as soon as you can.
I was actually quite worried about that. But, you know, having my dad around is quite nice for them. You know, they see him and they're like, it's
monkey
important
see,
for
monkey do,
yeah,
right?
Well,
Yeah,
it's
they
important
watch.
for boys as well,
Yeah,
right?
yeah.
Because innately, you are the hunter,
Right.
right? And I mean, I think we should support that in boys and men.
Yeah, to be strong and but in women too, they should exercise as well, right?
Sure.
Yeah.
But you know, it's
so you still got that that kind of traditional
I do
mentality of what man strong.
I do. I think that children, I mean, I see it with my boys, even though I'm a strong woman and I don't have, let's say, personally that timid female, let's say, demeanor or aura about myself. But, you know, when you have a child, you see the machine learning, right?
Yeah.
I see my children, or I saw them from the day they were born and have tracked their evolution.
Really?
And I can see naturally
what just
what
like by
comes
observing or by like actually tracking like metrics on them or
a little bit of both.
Oh, right.
I do little social experiments on them.
Oh, really? Like what?
Like what? Oh my God, I can't even think of one now, but I do it a lot. Yeah.
Right. Okay. So
but it's important.
So they train. So they're already doing what kind of exercises are they doing?
Lots of sports, but like my eldest does like working out at the gym.
Right,
and with my dad.
How old? You said 9.
He's 9.
He's already gym?
Oh my
are
God,
you supposed to be?
Body,
like, really?
I mean, naturally, he's gifted.
Right.
The kid is naturally gifted. And his body is literally like a man. He's got muscles popping out of everywhere.
I thought that you weren't, I mean, I thought
your growth,
yeah,
say.
Is that true? Or is that just one of those
I have to be
urban
honest,
myths?
It might be an urban myth. I mean, it's always good to solidify. You're right. Working out also helps the health of your bones.
Yeah.
But if you work out properly, you do a lot of stretching. My father always did stretching with me. Sure. I mean, I grew just fine, and I've been working out since I was probably 3.
Really? Like what, weights and stuff like that?
Weights.
Wow.
Yeah, he loves
Since
weights.
you were 3. Oh, my God.
Yeah. And with all the
maybe
Mr.
I'll get
Olympias
my kids
over
into it then. See what? Yeah. No. So
as
it
long
is
as
good
you
for
do a lot of stretching, right, hanging and stuff like that.
So you've been analyzing your dad, who's this 69-year-old guy that works 70. Out twice a day, and you said, what did you notice
Cold
in his
showers for like 30 years, blah, blah, blah. But I was actually talking about myself. So even though I haven't worked out for a very long time, we checked my body fat and my muscle, and it was actually pretty insane. I was shocked myself, and as was Kyle. So I think my body fat was 19%.
Oh, wow. Is that just from muscle memory from just years of working out since a child?
I mean, yeah. And diet, right? Because you are what you eat.
Sure.
So if you get those things in from the beginning, you have good foundations.
Sure.
And then my muscle percentage, I think, was 26.
Okay. That's
so again, without working out.
You know what I mean? I
was really shocked, I can tell you that. It was really nice. So after a couple of sessions, I could immediately feel my strength.
Coming back.
Immediately. The first exercises, I was a bit rusty, and it almost felt like I was clumsy in my movement.
Right.
And then the next session, my body came back to it.
Right.
And it's important. Like a lot of things, it connects your brain as well. So there are a lot of these exercises that you should do even as an adult.
Yeah.
Like before a presentation, when you're preparing for something that you'll be speaking about, that's important, like you're on the spot.
Yeah.
Crawling is good. So you want to crawling. Any exercise that connects the left and right hemisphere.
So just if you're in the green room, just crawl around just get ready before you go on stage.
Just crawl around.
Yeah. Interesting. So
going back to the one system, right?
Yeah. So the one system, that was interesting. We were just speaking about that. So there definitely is a big problem, I think, in kind of medicine and the way doctors treat. They're very specialized in just one area, and obviously
huge.
Yeah,
I'm sorry to interrupt you, but
please.
I've been trying to understand what the hell is going on. I mean, I personally, I trust my doctors. They're in Greece. I always fly to Greece. They're just, I think this lateral thinking, because of the way that
more
things
there
probably,
than here.
yeah.
So you think they're more open, they're more holistic than here?
Yes, I mean, they probably, because of our culture, always having to find a quicker route because things don't function. You're not comfortable. You have to find solutions. And so that gives birth to lateral thinking, right? You have to, I don't know, you have to find solutions. So that kind of is an occupational hazard. It becomes part of your part of the occupational hazard, right? Always trying to find solutions because you don't have the answer right there in front of you.
Sure. So would you say the doctors there are kind of more knowledgeable about various parts of the body, more like specialists in multi areas rather than just like...
Exactly. Well, I mean, think about it. If you're in a public Greek hospital where half of the shit doesn't work, you don't have the right staff, as much staff as you need. You don't have, the doctor's assistant and the assistant to the assistant. I mean, there are all these job titles now that I've never even heard of or even knew that it existed. There's like the doctor, and then there's someone under a doctor that's almost like a doctor, but not quite a nurse, but between a nurse and a doctor, and then a nurse beneath a nurse that's not exactly quite a nurse, but an assistant to a nurse. It's all, just like I'm lost. When you are just you and you have a patient on the bed that is in an emergency state, and you have to do it on your own with whatever you have available, you're going to become quite crafty, right?
Right.
You're going to have to think on your feet, and you have to find solutions. You have to become creative. So I think, now this is my theory, but also my experience. And I don't want to come down hard.
Yeah, I think there should be continuous education of doctors, even if they've specialized in one field, because like you said, the whole body's connected. All the blood flows around from one organ to another. So even if you're a specialist in a heart specialist, and you're prescribing medicine for somebody for a heart condition, then you would think they would also need to know about what effects that heart medicine is having on the rest of the bodies, because you see so much of that, like kind of doctors treating one illness, and then it fixes the heart or temporarily props it up, but then starts to affect the liver or the kidneys and all that.
I think we need to re-educate on biology. I think a lot of physicians have maybe forgotten the biology, the science side of things. Because yes, you have an organ, a big organ, but that organ is made up of cells. And those cells do specific processes, and they use specific molecules. And I feel that we need to re-educate and maybe kind of touch on the scientific progress that has happened. So we had a seminar and soft launch last week for our stem cell center. And it was really important for us to explain what a stem cell is. Because we hear a lot about stem cells everywhere. Stem cell facials, stem cells, exosomes for your hair, for your this, for your that. And it's become like a very broad term when in actuality, it's very specific. And actually, you're not allowed to call a specific cell or anything a stem cell unless it satisfies 3 criteria. One being, does it adhere to plastic? So if it adheres to plastic, so for example, on a flask, right? If it adheres to plastic, great, you satisfied the 1 criteria.
Adheres mean what? It means like sticks to?
Yeah.
Right. If it sticks to plastic.
Yeah.
Okay.
2, does it express specific markers? So every cell in your body expresses these markers on its surface, on the outside, so kind of transmembrane, right? So it goes from, let's say, the skin of the cell, if you want the surface of the cell, it sticks out, and it has markers such as CD105. Just take it as a word. It's a molecule, right? And CD105. So the stem cells that we use, the mesenchymal stem cells, have CD105 markers on them.
Okay.
And they have CD90 markers on them, but they don't have CD45 markers. So if you don't have CD45 markers and you have CD105 and CD90, then you are a mesenchymal stem cell. So you have to prove if these markers are expressed. So 2. And then the third is what they differentiate into. So what cell types, because stem cells turn into other cells, right? They keep their population. So it's like the beginning of a cell, which is, let's say, kind of like a
stage 1 cell.
Yeah, exactly. Yes,
sort of baby
it's like a blank slate kind of thing.
Then turns into something. Yeah, that's what. Yeah, my basic understanding of it was something like that. But please continue. Yeah.
That. So mesenchymal stem cells are, what's the word? Multipotent.
Multipotent.
Yes.
Nice.
So you have like totipotent, pluripotent, and multipotent. So they can only turn into a specific cell type of their type. So the MSCs that we isolate in our center, they turn into bone cells. So, they turn into osteoblasts. I was going to say fibroblasts. Osteoblasts. Because you also have osteoclasts, but they don't turn into that. They turn into osteoblasts. They turn into chondrocytes, and they turn into adipocytes. There are a couple of more cells which are question marks right now, because of course this is all very new, right? So we're still researching a lot. We don't know everything, but we know we know a good portion, and we know enough right now to be able to carry out some pretty cool treatments.
So stem cells, from what I've understood, stem cells, they seem to come, like we have them in our body anyway, right? So our body's always creating new cells so there's some part of where are they in the body? Where are they starting? And they is it like a sort of funnel they go through? Baby cell turns into now skin cell or hair cell or whatever.
We have a fantastic slide that really puts that into. The thing about science is that sometimes it's better to, a picture. Says a million words, literally.
Well, we could pull one.
It's just much easier to
show, right?
To understand. Okay, but there's different stages.
Are they all over the body though? Or?
They are. So the ones that we deal with, they live between the space of adipocytes, so fat cells.
Right.
So literally right between, let's say that's a fat cell and that's a fat cell, they'll live in this space.
Okay.
Amongst the adipocytes.
Okay. And they're the ones that are then sort of churning out these new cells as your body.
Well, they're immunoregulators. So what they are, let's say, just for a simple understanding, they're the firefighters of your body.
Firefighters.
Yes, firefighters. Right. So if there's inflammation somewhere, right, your cells communicate with each other. Like, you know how we'll have a text message, or back in the day, we would send letters to each other.
Yeah.
To be able to communicate with each other. Yeah. Well, cells will send out signals. And these are in molecular form. Right. So they'll be like, okay, I need this to happen. Send out a signal. The other one picks it up. Like, okay, we need this to happen. Can you make that? Yes, I can make that. But that process happens with a molecule. So there's several mechanisms of action of MSC. Some really cool images were published last year. I mean, we've kind of known, but it really kind of came out end of last year, I think. Really nice publication with some beautiful images of an MSC donating its mitochondria to a damaged adjacent cell. So not only was it giving it, let me back up, because there are, again, several mechanisms of action. Not only does it go to the site of inflammation, it also gives off these exosomes. So exosomes are these bubbles, let's say, that are stuffed inside with information, molecules that tell the damaged cell how to repair itself. So they give off these recipes, let's say, and then they also donate mitochondria, literally connected with a straw, and you see the mitochondria migrating from the MSC to the damaged cell to help it repair itself. And then the amazing part that you get, especially with autologous stem cell therapy, which you wouldn't be able to take advantage of with allogeneic, which is stem cells from somebody else's body.
Yeah.
You wouldn't be able to get tissue regeneration. So they pick up the signals from the cells that are around that point of inflammation. So let's say you have the knee, right? And you have osteoarthritis, and you have the cartilage that has been degenerated. So you have a little bit of cartilage here, but there's a pocket where you don't. So the cells, the chondrocytes that are still present will give off signals and saying, hey guys, we need you to turn into more chondrocytes. We need you to turn into us and start building out this hole.
Right.
When it's your cell, your cells speak your language. My cells don't speak your language.
Right. Even if you're like close kind of daughter or kind of relative.
Better.
Right.
But not entirely.
Because that's why, I mean, at least that's the rationale.
Well, it depends.
Behind
Don't
things
forget.
like saving your umbilical cord cell. Is that all just baloney from your...
Yeah, I mean, most scientists have not done that.
So when a child is born, you should just let it like pulse and then fall off naturally.
No, not fall off naturally. When it stops pulsating, then you cut it.
Then you cut.
Yeah.
Okay. Right.
Yeah.
So and there's no benefit of actually saving the umbilical cord and freezing it and paying annual fees for it for like
a lot of these companies have gone bankrupt.
Oh no.
Yeah, I've heard a lot of people, they've like lost their samples, and we don't have such long data on how, like if you did it 30 years ago, the data on the viability of the cells is not great, obviously, because this is something that we just a couple decades ago started.
So I'm surprised these companies are allowed to even offer this service then.
No, but the thing is, we haven't actually treated anyone with those cells yet. Not a single
everyone's
person
just saving them, and nobody's used them yet.
Yeah, no one.
Really?
We're not allowed to. Not a single person.
And that's
as far as I know.
Sure, and that's partly because in the UK,
there hasn't been.
There hasn't been. So like you guys are the first GMP-certified autologous stem cell clinic in the Western world, right? So you must have gone through lots of regulatory.
Yes, we have, and we still are.
And now you've been authorized. So you're like a pioneer in that area then.
We're in the process.
Right.
We're in the process.
So there's nowhere else in Europe or
and not as far as we know. Not as far as we know. I mean, we have to go not as far as we know. I know that RFK would like to start that process now, and that's going to take a little time for someone to build a facility like this, because we have built literally a GMP, so good manufacturing practice. And honestly, that acronym does not, it doesn't sound as intense as the process actually is, but it's a very, very intense process.
That's because you have a clinic and your own lab.
Yeah, a drug manufacturing facility.
Okay.
Excuse me.
Okay, right. Wow.
We have 3 clean rooms, but why I say drug manufacturing facility, although it's not a drug like a chemical drug, it is considered a cell therapy drug. So it's an ATMP, an advanced therapeutic medicinal product.
Right.
So because this is where medicine is going now, right?
Like more personalized. Yeah,
it's
which makes sense.
Personalized.
Which makes much more sense than
it does.
just like one treatment for everybody's
but
bodies.
we needed that as well. It's like what I say. It's, a culmination of thousands of years of science, right? And every part played a role. We had chopping people up, trying to, fix the knee and the heart and open heart surgery, breaking into the thorax and we needed those steps, right? Those steps have brought us to a point now where with our knowledge, just based on our knowledge, scientific knowledge, we can put that into play, right? Like everything that we do is based on scientific research. So things that we've seen in the lab, things that we have investigated over the years. And now we have reached the point of finessed medicine. So we don't need to break into your knee, right? The capsule of your knee. We don't need to do that. It's a very quick process. I'm actually patient 1.
Are you?
Yes, I am. I have to put my money where my mouth is.
Sure,
right?
yeah. Put your body where your mouth is, I guess.
Exactly. A little punch biopsy, 5 millimeters, right, kind of behind the ear.
And there's no scarring or anything from that.
Can you see anything?
Not really.
No,
No.
exactly.
So just like what? Take a bit of skin from
A
there.
little, well, skin. We want to get the fat layer, right beneath the dermis,
just under the dermis. Okay.
Yeah, where the MSCs are hiding.
Okay.
So we take that, and well, what we've done, which is very unique to us, we took the regulator's advice, and we thought actually that'd be really cool for many reasons. But everything is under one roof, so we control everything. We can monitor everything. Nothing gets lost.
Sure.
We have full chain of custody. We have built 2 operating theaters adjacent to the clean rooms, so the labs, right, the drug manufacturing facility, which are connected via a pass hatch. So they're these like
little
boxes
kitchen hatch things, is it right? Those through the kind of.
Well, kind of. Let's go with that. Not exactly, but a little fancier, with HEPA filters, like cleans the air. I mean, it has to be sterile. You push a button.
It's almost like a spaceship or something.
Yeah,
like,
yeah. I think a lot of people have kind of described our facility as out of a sci-fi movie.
Really?
Only that it's not
I'd love to have a tour.
It's sci.
Yeah.
Oh yeah, you haven't been. You definitely need to pop in. So we take that little sample, we put it through the pass hatch, and then the process begins. So it's literally, the sample goes from patient into the lab directly.
So it's like a mini surgery. You're on like a kind of, what do you call it?
Yeah, we do everything a little like over the top.
Right.
We just, we are trailblazers.
Yeah, that's
right?
what I was going to ask. I mean, if you're the first to do this, you're creating the model then, because you've got no model to follow. Or are you following any other countries that have already authorized it?
Well, so my business partner is Dr. Takamatsuoka, who has been doing this in Japan. He was the first to do it to get the license to do it in Japan.
Right.
The Japanese actually got the Nobel Prize, Yamanaka, for some really incredible things in the stem cell space. He turned a fibroblast into a stem cell, and that back into a skin cell. Now, I'm definitely saying that process a little backwards, but he introduced some factors, which are now called Yamanaka factors, named after him.
Wow.
Which were able to convert a cell back and forth.
So this Nobel Prize winner is your partner in the business?
No, he's not.
Oh, yeah. Oh, yeah.
He's not. But he should.
He's the one that pioneered it.
So he's the one that, together with several professors at the university there. Now I've blanked which university it is. University of Tokyo, I think it is. Well, excuse me while I check my notes. Sure. But I've had a very long couple of months.
Right.
So the government, the regulatory body in Japan, allowed stem cell therapy to kind of happen. So they created a special department to push this, let's say, technology forward. They're like, you know what?
Yeah, let's try it.
This is pretty amazing. Let's try it. And we were the first.
Why is there so much pushback in the West on it? I mean, obviously,
we're a little more
injecting people with cancer is not, you want to avoid that.
Yeah.
But if it is so revolutionary, why aren't the governments really behind it and pushing it? And it feels like they're doing the opposite.
Yeah. There are reasons. I mean, look what happened with the vaccine, right?
Oh yeah.
They pushed it forward, and then everyone's like, "Well, why did you push that forward?" There are arguments on both sides. That's 100%. But
the vaccine was kind of being forced on people, but this seems like obviously people would elect.
That's true.
To do it.
That's true. It became
but they're being blocked from being.
It became forced on people.
Yeah.
But you do need to be careful. You do need scientific rigor. You can't just plunk any, new crazy science or idea
into humans.
Exactly. So there are cases for both, but I heard someone say it perfectly the other day. The problem is it stops innovation. When you're being too careful and too regulated, it can stomp regulation. But then on the other hand, there are a lot of cowboys out there.
Sure.
A lot of cowboys. I mean,
underground stem cell clinics, like, hey.
A lot of people that claim that they're a stem cell, that they do stem cell therapy, that we've been looking we've been looking into, and it's criminal. There's someone on Harley Street and a surgeon, an orthopedic surgeon, that claims that he does stem cell therapy. And what he does is aspirates bone marrow out of the patient and to collect stem cells. Everyone loves this word centrifuge. Do not get impressed when you hear the word centrifuge, please. For anyone that's listening to this, centrifuge just spins a sample and separates just the size of like things that are in that sample by molecular weight, for example.
Sure.
And you need to know how to spin things because you could be lysing the cell, so like breaking the cell. It's not, I don't know, rocket science. It's not, and it's not an instrument that's so sophisticated. Anyway, so then he takes this sample. He takes something from the sample. I don't know, one of the phases. I don't know where or what. He probably doesn't know where potential stem cells are. And then he reimplants it intravenously. Now, stem cells that are in the bone marrow, that's their job. They go from inside the bone marrow, inside the bone, into the blood, from the blood, into the bone, from the bone, into the blood. That's what they do.
Right.
That's what they do. So what exactly, what has he actually done?
How's he allowed to do that then?
He's not allowed to do that. But, I mean, it has been a gray area because, again, it's very new. But I think the MHRA, which is the regulator here in the UK, it's the FDA of the UK, will be cracking down on these people. I mean, how can they even keep an eye on all these cowboys? But I bet you that this orthopedic surgeon probably doesn't even know what a stem cell really is. He probably doesn't know that the scientific community in stem cell research has actually given these, sets of categories that you have to satisfy that we discussed earlier. They just hear this, this word. They don't know the science behind it. They're not really, this is going back to what we were discussing before. It's just the end treatment. But what a physician is treating with is the research that we've done, right? It's kind of the end of the life cycle of a treatment.
So they administer it. So you've done research.
Yeah. This is our research.
And now you're applying that research into practice. So does your research, did it involve human trials or anything like that?
We've treated 16,000 patients in Japan. So we are in a very unique position.
So that's quite a big data set to show that. And
that's why we're allowed to do this.
Solid results then.
Again, let me
So what kind
track back.
Yeah.
That's why hopefully we will be allowed to do this.
All right, so you're not 100% authorized yet.
We are not authorized yet, but we have everything kind of ready to go.
So
because we've built the facility. We've been speaking with the regulators. They've been actually extremely helpful and really nice. I mean, I think, this new blood of MHRA agents are just phenomenal. They really are. They work with you. They are your partner because that's what you want. You want to make sure that you're doing everything right.
Sure.
And you want to make sure that there's oversight from them. You're not scared to speak to them. You're not, the old house, I think, was a little bit more like austere and like, I'm sure there's going to be, a little more. It was a little bit like
adversarial as opposed to collaborative.
Exactly. That's the way. That's exactly.
So of the 16,000 patients, what kind of things can you treat with stem cells, and what has it been shown to definitely cure or help?
No, we never say cure. In science, we never say cure.
Okay.
Never
or
cure.
Like improve or whatever. what kind of
therapy? So what
is
indications?
It a preventative? Is it a kind of?
Oh yes, you could definitely. You can't treat in the Western world. You can't treat for wellness. That's not an indication. An indication is a disease area because you kind of have to show
something's broken first.
Yeah, it has to be broken first. Right. But you know what the beauty about these stem cells are? You just reimplant them and they go off to do their thing. So there are things that you didn't know or would have never known, but they're speaking to all the cells on the inside, right? They're speaking to all the different cells in your body. So if they pick up that there's inflammation somewhere, which has not been detected, has not reached the point where there's damage, like proper damage, where you start to feel the pain, they'll just go in and fix it.
But if these stem cells are in our body already, what is the therapy then? Are you just like adding more of them? Or?
Yeah, it's more targeted. You're boosting it, right?
So you're just suddenly just like reinforcing.
You're reinforcing
the sort of to help in that area. Because if we have it already, why aren't we just always self-healing and?
Well, we add a lot in. Right. But there is, we can go down the deep science of it. Actually, Dr. Cristina Filipeos, who is our CSO, she's a stem cell expert. She's phenomenal. She's got 2 patents under her belt. She's been doing this for years, day in, day out. And she was the right hand of our other partner, Professor Fiona Watt, who is the mother of stem cell biology.
Right.
Yeah, so she's also one of our partners. One of the things that Christina discovered was, so all these cells kind of have subcell types. She found these subcell types. And she found that when these cells are together with other cells, subtypes, the signals can get drowned out. So again, our bodies are not perfect. I know everyone thinks that they are, but they're not.
So right? The communication of, hey, there's inflammation, come and help us. That message isn't getting through.
For a specific thing, right? Because there are subtypes that do specific things. Like there's a subtype of fibroblasts that produce collagen, and then there's a subtype of fibroblasts that does something else. When they're all together, they seem to drown out kind of each other's signal. Again, this is something that she should be talking about.
Okay.
But
it's like too many cells trying to do too many different things at the same time.
If you reimplant a large number in a specific area, then the signal will be strong of that specific cell.
Right.
So to isolate those cells, you'll get a better response.
I see. So all these women that are obsessed with collagen, can you do some stem cell collagen treatment then?
Yeah, those are fibroblasts, yes. Well, I've saved my fibroblasts.
You've saved them?
I have, yeah. Not just my MSCs.
Where did you extract them from?
From the same exact biopsy that
From that one little thing. Then you can multiply them in the lab? Right.
Yeah.
And then can you,
I haven't done the treatment because we do nothing that's not allowed yet. We need to
even
get
when the
all
doors
the
are closed and there's no regulators around. Quickly, inject me.
Well, actually, my dad was like, my knee, I can't walk. Can you please? I'm like, he didn't say, can you please? He was like, when are we going to do my knees? And I'm like, Dad, sorry, but...
Rules are rules.
Rules are rules. We're not we do everything above and beyond. Literally, because there are guidelines.
Sure.
Right? There's the Annex 1 that we follow of the GMP guidelines. And they tell you, okay, you have to have, for example, you have to have HEPA filters. We have ULPA filters. So, a step up, the max, the top. Particle contamination events.
What's that, air filter? Is that type of?
Yeah.
Right.
So we have positive air pressure. We have cascading air, like clean air has to always go from, because we have 3 clean rooms, grade B, grade C, and grade D, which have different pressures as well. So grade B has 65 Pascal, grade C has 55 Pascal.
Pascal. What's
Pascal
that like?
Pressure.
That's a pressure,
yeah.
Right.
And then grade D has 45 Pascal. So you have, the gradient. Always ensuring that clean air goes that way.
So when it's clean, then that's clean to what level? Like there's...
very
clean.
0.05 micrometers. Yeah, so you're cleaning out particles. Right. And we have sensors in our ceilings, and we have 2 meters of tech in our ceiling. We measure everything.
So that's to make sure there's not a single bit of bacteria or viruses and what
everything.
Everything's filtered
particles.
Out. Right,
just
particles.
Generally, yeah. Wow. We have an environmental monitoring system. So every half a meter of our lab space, ceilings, walls, floors, touch points, everything is monitored. Everything has a name. Right. And then before we start working, after we start working, weekly, daily, quarterly, and monthly, we take swabs. We have these agar plates, so you can do kind of touch a specific area. Then you record it before and after your cleaning, before and after your process. And then we send it to a third-party lab. And they tell you if it's been cleaned, what it was like before, after you did your cleaning process. We also have to validate our cleaning. And just monitor your day-to-day in there. So when we're working in our hoods, where there's a grade A environment, you also have a settle place. So you have like these Petri dishes. You remember Petri dishes?
Yeah, so
with
a
agar?
hood is what, like your clothes? What?
No. A hood is your biological safety cabinet. So
okay. Oh, so you're wearing what, like
oh my God, your
suits? Is it
yes,
one of those proper?
but it's the space.
Because I was
going to
ask, like, what about when the humans go in? Aren't you guys?
Oh no, we're fully hazmated.
Oh, you're hazmated. Okay, so you are. Yeah. Oh wow, it's that.
Oh
level.
No, it's like it takes me personally. It takes 25 minutes to get suited and booted.
Just to go in those rooms.
Yeah,
wow.
and everything is like gamma rayed.
Gamma rayed. What's that?
Gamma rayed. Gamma.
What's that like?
Like, you know how you have X-ray, gamma ray.
Yeah.
So you just like kill everything. So they package it and they put it into one of these, I don't know, I don't know exactly how they do it, but they gamma ray it into like a special.
And this level of scrutiny is to make sure there's not a single particle entering into the Petri dish, which then goes into.
No, the Petri dish is just to monitor.
Okay.
Just so we know what the environment is like.
Sure.
It's so that nothing goes into your sample. You don't want your contaminated sample.
Go into the body. Sure. Okay. Wow. Interesting.
Yeah.
Wow, that sounds intense.
It's so intense. It's so intense. We have to clean floors, walls, ceilings, every nook and cranny. I mean, everything is built very specifically, you know, because you don't want like corners, crevices. You're not allowed to everything has to be flush.
I see.
I mean, the materials that you use, I wanted all of us, you know, people that are in the lab, all the scientists, not to have to wear polyester because I hate polyester.
Yeah,
you know, I mean, it is petroleum at the end of the day on your skin. And I was trying to find something that was a healthier option for our manufacturing scientists. And then we found out that you can't because cotton or any natural fiber sheds a lot. So it has to be polyester.
Oh, no.
And you have special, because we wear layers of things. We have 2 layers of gloves. We have goggles and masks. I mean, nothing is out. Special gloves.
My God.
Booties and special shoes. And then the coveralls where you have to step into. We have like what's called the step over bench, which separates the clean side with the quote unquote dirty side. So when you're in the dirty side and you're walking every step of the way, when you go in from the grade D, again, remember we said cascading, we have 2 airlocks where there are different stages of our gowning. And we have an SOP. We have a protocol, standard operating.
Oh, standard
procedures,
right.
Yeah, so all of that is written down.
Right.
So there's a specific way.
Each time. Wow.
Yeah.
Each
there's
time
like
you
35
walk in, then what are you getting, like sprayed like like in the
air
movies?
curtain.
Like...
No, we don't have that. In the West, you don't really need to have that. That's in like category 3 or like 4 for in like places that study viruses.
Sure. Right.
But not in this scenario. So every step of the way, you have a specific way of how you gown up.
Sure.
And the different stages. Like we actually we write them down. And
step by step. Because if someone gets casual with it, then
you can't. Everything is very specific. Where you grab the thing when you not unfold, when you unpackage. Oh, and also, by the way, everything is triple bagged.
Triple bagged.
Yeah. So every step of the way, it take 1 layer out. The closer that you get to the grade D. And all of this, by the way, is submitted. You include that in your dossier that you submit to the regulator.
For every patient, every time?
Our process.
Right.
But the regulator as well. So we have a LIMS system, which is a lab information monitoring system, which from the moment the sample is collected, then we have another system that actually is for the patient itself, but for the sample, it has a barcode, and we have site and auditable, traceable information of that sample throughout its lifetime. Who's had it, what time they had it, as in, you know, our manufacturing scientist, what, let's say, ingredients were used, just to make it a little in the processing of that sample, the batch number of that, the kind of the birth of those ingredients, or the chemicals. When we say chemicals, we don't mean it in the sense of like, you know, how you would use it in everyday lingo, but like the substances, let's say, what has been in that, where those things came from. I mean, the audit trail goes back to the birth of every single thing that has been included in everything, like and that's, our instruments as well.
That's made by the regulators then?
No, we make that, and then the regulators have oversight, and it has to be GMP.
But who designed that protocol? That was you, or that's following the Japanese system?
So that's not just
system.
us.
Right.
No. That's everyone has to do that, and it's
that's
us
law.
For our process, and then also our vendors. So we've partnered with Thermo Fisher, which we're very honored to actually have them present us to the world and back us. They've become like a family, and I think they also feel the stress and the joys and all the roller coaster of emotions that we've been through. They feel it with us. They have just been amazing. Thermo Fisher Scientific is a huge, I call it science everything company.
Okay.
So they do pretty much everything. They're like a $200 billion company.
Wow.
Yeah. And actually started by Greeks, but it's an American company, and they have just been incredible. And the substances, let's say, the things that we use, be it instruments or reagents or, you know, anything in the process, they actually make. And those are specific to GMP manufacturing. Right. So they have this, like, we have to IQ, OQ, PQ, DQ, which are different qualifications, like installation qualification, operational qualification, design qualification. There's like all these, you get literally a file just for the one instrument.
Oh
they come in with engineers, and if you move that instrument, then you have to do it again. So if you move it from here to there, you have to do that process again.
Oh my God.
Yeah, I mean,
wow. Sounds like a huge audit trail and supply chain audit going on.
There is oversight on everything that happens in our facility. And that is important. Like, I don't feel bad about that. I don't feel like
you don't want to leave any room for errors in this kind of stuff.
No. And the thing is, you know, for me, like, what solidified, or as I said, crystallized my this undertaking. I built it for my mother.
Oh, really?
Yeah. So my mother was diagnosed with ALS.
Okay.
When they call it MND here in the UK. And for me, it was really a race for her life. Dr. Matsuoka Taka had offered her treatment in Japan, but, you know, with ALS, you're completely paralyzed. You lose control of your muscles, right?
Is that a gradual thing as well?
It's a gradual thing, but it's pretty fast.
Oh, really?
So
it's not like MS that can take years, right? No, it's a lot faster.
No, it's like prognosis is like 2 years, right?
Okay.
Yeah, a year and a half, 2 years. It really depends. Like, I don't even know how Stephen Hawking continue for so long, honestly.
Right.
I mean,
so
he's the only person that, that's what he had. And that's what my mother had. It's really a miracle, but it went fast with her. So we quickly realized that it wouldn't be viable, you know, from Greece to go to Japan. There is no direct flight, so there would have to be a layover. And then how would she go to the bathroom? She was not at the stage where, you know, she had a catheter or anything like that.
Right.
And you wouldn't, it'd be difficult, you know. It was kind of not in the early stages, but she couldn't walk to go to the bathroom if she needed to. Sure. And then what do you do? I mean, it would have been too much.
Pragmatic,
and it takes a long time, so she'd have to stay in Japan, and I have 2 little kids, so I wouldn't be able to stay with her so long there. And So I quickly realized that, I needed to get moving, and it was a race for life. So we started that process here. Unfortunately, she passed away before she could benefit from it. But there are other people that were helping.
So you thought because you couldn't get her on a flight to Japan, just set up your own clinic here. That's a pretty big undertaking as a solution. How about private jet? Oh no, can you just done that?
The thing is, she'd have to stay there for it's
the
long
staying
process.
there. Is it a long? It takes a long. So and has it been proven to help then from the previous patients?
There is promise. There is promise. There is promise. The therapy generally shows a lot of promise in the neurodegenerative space. Exosomes.
A friend of mine had MS, and I think he was going to Canada to do stem cells. Are they allowed it to do it there? Or no? Or maybe someone that was calling it stem cells treatment.
No, maybe he did. Was it maybe a clinical trial?
Oh, maybe, yeah. Or maybe a clinical trial, right.
Yeah.
Okay, so they are doing trials, but they haven't, right. But in Japan, if they've got such evidence and it's kind of proven, or, I mean, that's...
Don't forget, it also depends on your cells. We can't predict it. But if you read a lot of the research,
mixed results, or
generally it shows a lot of promise.
Okay.
I'd have to show you the data.
Right.
We don't like making big claims. We're not doctors. We're scientists.
Okay.
We don't sell.
They would never. Sure.
Yeah, but
so
really nice studies out there.
Right.
And again, it really depends on your cells. Like for example, we took a couple of samples for our validation runs, and we've been taking care of our stem cells. We've been, we use microscopes to see how they're doing.
Yeah. So once you take the sample, are they then? How long is it in between you sample and add your rest of your ingredients before you start injecting, and how many times you?
You don't need to do it. So with allogeneic, because they're not your own cells, they actually get eliminated from your body after 48 hours. Oh. I mean, it's a simple solution, right? There's in a few cases, does allogeneic stem cell therapy make sense? If there's a specific mutation in those cells that need to do a specific process, but there aren't many indications where you would need that.
And that's from someone else's.
Someone else's, yeah.
So
and we all know
you don't believe in that. So what you're doing is autologous.
Autologous, your own. Yes,
there
exactly.
You go, which means your own cells.
Your own cells.
And that's the one which you reckon that is going to make the biggest difference.
Yeah, 100%.
Right. And how long?
We know the mechanisms of action. We don't really know what happens with allogeneic. They call it hit and run.
Sure.
And that's just kind of like, I don't know what it does, so hit and run.
So your patient 0, you've taken out your own stem cells from your neck, and then what happens? And it's like you go through that process. Now, is it like on ice, or it's
on ice? It's in liquid nitrogen. So we also have our own cryogenic storage room.
All right.
We have our liquid nitrogen tanks
there, and then what? Then you can multiply it in the lab somewhere before injecting back.
Yes. So there are some parts of the process that I can't reveal.
Oh, okay. Oh, really? Trade secrets?
Well, there's some trade secrets.
Okay.
But we do multiply. We allow them to multiply. And we also have patented our own media. So media is the food for the cell. So all the wonderful things that your body would feed them, we give that to them, right?
Okay.
To live. All the nutrients, et cetera, et cetera. And we have some really amazing data on that.
Right.
Anyways.
That's kind of weird, keeping cells alive outside the body. It's like crazy. Yeah.
Yeah.
And are you doing any like growing organs or anything like that? Because people speak about, that's part of like a kind of shoot off that, right? Off the stem cells that they're people putting it on scaffolding and are they trying to do that? Is it?
They are trying to do that in Germany. You've read about that study, right?
Yeah.
That group. Yeah, they are. And that would be absolutely fantastic. I don't think we're yet at the stage where I don't think it's been proved yet.
We can grow a liver and replace a liver and stuff.
Yeah, with the scaffolding.
I mean, they're trying to do some stuff like that in the kind of vegan alternative meat, or where they take one cell of an animal and they kind of multiply, and they're doing
Yes, and I actually know of a group that's doing that. But again, it's difficult right now. So Thermo Fisher is working with some people that are doing that. And I mean, it's really fun to see the science. And that would be also really cool, right?
Just
you
to
could do
grow
that,
meat
but it's still very expensive
suffering.
to produce.
Yeah.
Like a kilo of that meat.
Yeah. Right.
But we're getting there.
Lab
what was it
meat,
when it first? Yeah, the lab grown meat when it first came out. What was that? It was like
I think
I don't know how much.
I don't think there's many governments. I don't think there's many governments that have authorized it. I think. I know they had it in Singapore. I think they've actually serving something like some lab grown chicken like into some kind of nuggets or something.
Really?
Yeah, because we made I
didn't hear about that.
Into one company called Good Meats that's done something like that, part of the Just
but
Egg.
How do they get the pricing down? Because I mean, you need to do it in really special environments.
I mean,
and then you also have to make sure that like things that you had used, like for example, trypsin, that there's no traces or an acceptable range of trypsin in
trypsin as well.
Trypsin is it's an enzyme
that can be damaging to you.
Funny enough, we were looking for some papers. It was very difficult to get any information, which usually, you'll have a couple of papers on a specific subject. I think like any kind of deeper research that had been done on trypsin was like from the 50s or 60s, something crazy. But there's an acceptable range. I mean, you have that, it's pretty standard in our processes. But it cuts specific amino acids, like glycine. The protein, it'll cut it in a specific place. It reads kind of, let's say, the code and the amino acid sequence of a protein, and it'll cleave in a specific area. And
that's good or bad? Or that can...
I mean, we use it to benefit us, but
But in food, it could be bad?
If you have... I don't remember what the allowance is right now off the bat, but you don't want to go over a certain amount because it can cause problems. But usually you want to do more washes of the trypsin to make sure it comes out, because I mean it's an enzyme that cleaves, right, that breaks up protein. So you just need to follow the rules. That's the thing. You have to get it into the levels that are acceptable.
I see. And that's...
that whole process is, it's very expensive.
Right.
So I'm surprised that they've already...
Yeah, they must have come up with some solution. I mean, I could probably look it up. But in terms of what treatments you can do then, like what's going to be on your menu in terms of like what kind of clients are you trying to attract and what are you offering them?
So our specialist license is in osteoarthritis. That's what we're going for. Very direct. We want to start small. In Japan, we have 15 indications. So for 15 different disease types.
Okay.
So
you're linked to this Japanese clinic, or you're kind of feeding off their...
No, we're total. It's my business partner is Dr. Taka Matsuoka.
Okay.
So we're doing this together.
Right. Okay. So they're kind of just taking the model there and putting it here.
Yes. I mean, there are changes. There are differences because there just are. Because of the nature of the regulators here, right? There's different regulations here. There are a little stricter rules, let's say. Like it has to be a GMP lab here. It's not in Japan. They have ISO. And just the processes here are a little bit different. So we have to follow those. And we have our own experience with like cell culturing, which is basically what we do.
Sure. So you're starting off with the osteoporosis. That's like arthritis.
Yeah.
Arthritis.
And then what we can do generally, kind of what's part of the allowances from regulators generally is a pathway called named patient. So generally what that means is if a physician in one country where a drug is not authorized, they've read, let's say, studies of that specific drug, and they have seen that there is some efficacy that they think would work, this drug would work for their specific named patient, and that drug in another top-tier regulator in a different country has licensed it, for example, Japan, or Korea, or Canada, or, one of these respected regulators, then that physician, and it's the responsibility of the physician to prescribe that drug to his or her patient,
that
the
particular
specific
individual person.
Exactly.
Right. So it's
That's
not
why
like
it's called
a general
named patient.
I see. So it has to so you have to get like a pre-authorization for each case. You can't just offer it blanket.
It lies on, it's the responsibility of the physician. If he thinks or she thinks that their patient will benefit from this drug, and don't forget we're considered a drug, even though we're not in the traditional sense, we're a cell therapy drug.
It's like customized.
Exactly. Then they can put that order in, let's say, that process to us, and we will produce that drug, the personalized drug, for their patient, and then they can reimplant it.
And has anything gone wrong previously, or stuff has gone wrong? Just looking at that.
And everyone,
devil's advocate.
My motto: we're safer than aspirin. So out of these 16,000 patients, we have never had a single adverse event greater than a grade 1. A grade 1 is fever. Nothing. Okay. So that's
no
really
like
unparalleled.
Frankenstein morphing. Whoa, we put the wrong.
And in Japan, under their regulation, the PMDA, their regulator, which is the FDA for Japan, they require the physicians, every physician that does the therapy, every year, their audit, for example, they need to present the data of all the patients that they have treated in that year. Right. So all of that data,
make it public.
It goes to the regulator, and they submit it, and they go in front of the regulators, and they present all the cases that they've treated.
Nice. And
so we're in good hands.
So that data sounds pretty useful. I mean,
very useful.
Have you looked into the use of AI or any kind of processing that? I mean, in your work?
Yes. So we will be doing some fun stuff, and this is kind of the exciting aspect of our work, of having everything in house. Right? We can literally do research, delve into one aspect, something that we might have noticed, something that we saw in someone's particular cells, or something that we just thought of, it'd be interesting to see how that links to that, or what happens when you do this. And we can do it ourselves in our own lab. It's quite fun, you know, but it also speeds up the process. What I like to say, as you would say, what we do is farm to table, from bench to bedside.
Okay.
And I think that's also something that's missing, in medicine, because there are so many, again, what we were saying, you need to be regulated, and you do need to have these strict rules that you abide by. And that's what we've done. We've gone above and beyond. Even our operating theaters are built out from grade B panels. So you just need to have grade B panels in your grade B clean room. But I've built the whole lab and the operating theater out of grade B panels. I have ULPA filters everywhere. So we've gone above and beyond for everything to ensure safety for our patients and quality of our product. It's very important. So we have set that foundation, and then, we can become creative and do our own R&D, which we do, and we love it.
Sure.
I mean, we really do geek out.
Right. So that's what you had done
in our center.
Before this. You had done a lot of research.
Yeah.
Where was that in like Japan? You gone to, or where had you studied or built up before?
Yeah. Well, I'm actually still at my PhD has been a collection of Cambridge, King's, UCL, and Manchester.
Oh wow!
And
I didn't even know that existed. Like a multi-uni PhD.
Well, you in science you can do that. You do your I've done wet labs and dry labs. Now I'm doing dry labs. So computational work. My PI is Professor Franca Fraternali. She's an incredible woman. I owe her everything. I really do. She's a veteran in the space. I mean, the biophysics side, the computational side, is something that is starting to gain a lot of attention. Last year, our area received 3 Nobel Prizes. And it used to be that, we were the underdogs, and it was just kind of like in a research paper. You do research and then you publish it.
You received 3 Nobel Prizes?
Not me.
Oh.
No, not me. Our area.
All right.
Our area.
Oh, that area. Okay.
The area, which was the underdog. They were the we were the underdogs, and you wouldn't actually just publish based on just the findings of the computational side.
Oscars, like in this year's section, we have the finalists and that. So,
well, it's big, right? Because it's our work collectively.
All right, so you don't know until the award ceremony who wins.
So the Nobel Prize.
Yeah. Is it a bit like that?
They tell you before.
They tell you before. Okay,
yeah,
so you get your speech ready.
But again, I'm not a Nobel Prize laureate by any stretch of the imagination.
Yeah.
But
Yet, right?
Well,
yeah.
Let's just concentrate on what we're doing now. That's
sure. Well,
it's a big undertaking.
And sort of pricing, is this something? Oof.
Yeah.
Can you divulge or no? Is this something which is going to be available for everyone, or is it going to be available on people's insurance, or is it going to be just like,
we're
privately
taking
funded?
Things step by step. The next step would be to start discussions with insurance companies. Definitely ones in the U.S., I think, would be the ones that would be more interested initially, because, the treatments in the U.S. are very expensive, and just cost-benefit analysis, just if you take the life cycle of like someone, for example, that has knee osteoarthritis, and I guess the pathway will always end up in having knee surgery.
Yeah.
If you just think about how much you're spending on from the first moment, right? Like getting it diagnosed and having physio, then these injections with arthrosamide or hyaluronic acid, or all these different therapies that is used currently, but really no treatments. And then having surgery, does it actually make more sense economically to do stem cell therapy?
Yeah.
And for me, I think it does. We're crunching the numbers as we speak because, again, we're a lean team.
So you don't have a pricing plan yet for what you're treating?
We do.
You do? Right.
We do, but
it's not public yet.
It's not public yet.
Sure.
It's not public yet.
Yeah, and then how long does it take? Is it like a one like sample, grow it in the lab, inject me, and then I'm done, or do you have to go back and multiple times?
So the whole process, and I've chosen to do the cleaner process, so we don't use more ingredients than we need to, because again, as I was telling you earlier, I believe in organic. I want things to be clean.
Okay.
And whatever I do in the center is what I would do for my mother, for myself, for my children. Because every patient is a mother, is a child, someone's child, someone's sister, someone's brother, someone's father. Our patients are not just numbers. And we can never forget that. We want humanity to be a big part of what we do. And I think sometimes maybe scientists forget, or maybe people think that scientists forget. Maybe it's a bit of both. But humanity is number one for us.
Sure.
Science and humanity can coexist.
Yeah.
So our process, therefore, takes a little bit longer. And of course, depends on what's the number that we want to get to. So if you're doing a local reimplantation, that would be for a knee, for example. Yeah. That would be 100 million cells. But if you're doing intravenous...
100 million pounds. I was like, bloody hell. For a knee. Okay, 100 million cells, all right.
100 million cells.
Okay.
But if you were doing intravenous reimplantation, we like the 2 billion mark. 2 billion, we've done internal research, and 2 billion is the sweet spot.
Of where? So you reproduce 2 billion first,
and
then
grow into 2 billion and then inject it.
And that's just like one session?
One session.
And then you're done?
Yeah.
So you just go and take it out, and then you grow it in the lab, come back in a week or whatever? Yeah. How long does it take to grow your 2 bill?
Depending, 2 bill would take us a little over a month.
Okay.
Let's say 1.5 months.
Right.
For 100 million, we'd say just under a month. So 3 weeks.
3 weeks,
yeah.
Wow.
So in our process, there's, and again, through our whole process, we test the samples twice, so before we freeze them. Mm-hmm. You could come and, let's say, have your therapy. Like, we take the biopsy, we wouldn't freeze it, just do the process, directly. as in grow the cells and then reimplant them without freezing that first therapy. We produce 3 vials.
So you can have just cook them up and put them straight in, no freezing.
Yeah.
Right, for later.
You would for the one. The rest we would bank.
Right.
In our own facility.
I see. In case you want to go for like round 2, round 3. Okay.
Exactly. It's always good to have them.
Yeah. And is the promise of all this that it would turn into, one day, okay, my cartilage is all run out, regrow it, like,
you regrow it.
Yeah, like
we plant the cells
regrow
and then
it.
you
Like, would we?
it.
I mean, could this effectively one day is the trajectory of this tech heading towards kind of longevity and anti-aging and
you
like
can't treat for longevity. That's not an indication. However, as I said before, if you were treating your knee, or you were treating some other disease, then inevitably you're also
extending your life. Yeah,
exactly.
But can
treating longevity?
Right. So you can't because we're getting older all the time.
There's always something wrong, right?
Yeah.
So you're getting multiple benefits. Like we had one of our patients in Japan when we were looking at our data back in August, and because obviously we've got thousands of patients. One of the patients that we had, again, it's all anonymized.
Sure.
We do everything by the book. She came in, it was a she, for CVD, cardiovascular disease, and her blood panels were showed that she had really, really high liver numbers, like extraordinarily high liver numbers. She had the therapy done, the stem cell intravenous reimplantation, and her liver numbers decreased by 1,000x.
Okay.
I mean, it was just insane.
So back to being healthy.
Back to being very healthy.
Wow.
Yeah. So she came in for something else, and the stem cells fix something different.
That's cool. Okay.
So
let's have a look at an example of
what we've been discussing.
So here we have an example of a patient with osteoarthritis, and
it's an arthroscopic evaluation of a 30-year-old female.
Right.
And you can see right here, this is the cartilage. So chondrocytes right here. And here, this area is empty, as you can see. So that's degeneration. Now, 4 months post-treatment, which, by the way, 4 months is when we checked, right? So this could have been produced earlier. I mean, we have some other imaging results that have been taken at 3 months, and we still see that there's regeneration. So as you can see right here, we have new tissue that has been produced. Wow. So those stem cells have now become chondrocytes. Isn't that amazing? So literally the power of the gecko.
And then that will obviously get rid of all the pain then that they experience.
Yeah. So this treatment is really effective for people that have up until grade 3, because you need to have, remember those signaling molecules we were talking about before?
Yeah.
So you need to have enough signals that are released from the other chondrocytes that are still there to tell that enough of the signals to tell the stem cells to turn into this new cell type. For grade 4, you have so little cartilage that you don't actually get tissue regeneration. However, we have seen that according to the WOMAC scale, that you do have better functionality and much less pain. And I mean, pain is a very big thing, right?
Sure, absolutely.
I have osteoarthritis myself.
I do.
I do, yeah. So I am literally going to be patient 1.
Really? Wow.
Oh, yeah, I'm
waiting.
That's
good. Put yourself in the firing line.
Yeah. So, I mean, pain is it's a problem, right? Every move that you make, you have to consider. So
that's why a lot of people, I mean, is there any truth to people using things like CBD for arthritic, for pain?
Yeah, again, I think that's pain modulation, right?
Pain rather than actually healing.
This is actually treating the cause.
Right, which is just a kind of degeneration of the cartilage over time.
Yeah.
And what causes that?
I mean, I think it's mechanical, right?
Oh, just from use.
Yeah, it's from use. It's from use. But we weren't able until, until present to actually treat that, right? It's really only managing the
symptoms.
The symptoms.
Rather than healing.
And then, surgery, it's intense. And you're breaking into your knee capsule. And, surgery also doesn't last. And.
Sure.
So, who wouldn't want to just do a simple injection into the knee and literally just grow your tissue back?
And then it just grows in your own leg over time until it's back. Wow.
Mm-hmm. And again, I think the most important thing is that it's safe. Our safety profile is unparalleled. I love to say, and it's the truth, we're safer than aspirin. Sorry, guys. But, having just a grade 1 adverse event, which is fever, is really nothing. One of the things that medics, scientists have been afraid of is cytokine storm. We've never had a case of cytokine storm. There's no reaction because they're your cells. They're not a foreign body. And that's the thing about allogeneic stem cells, right? On the one hand, they have a paracrine effect and they are immunomodulating, but actually it's still a foreign body that's entered into your body. So there is a reaction, right? An inflammatory response. Again, we don't know much about it. Research has shown that there is a positive effect in a way, also in diabetes.
And that can be from anybody else's, or
so
they tend to use somebody close to you. Jeff, can you move around a bit?
Hold on a second. I want to show you the difference. So autologous versus allogeneic.
Okay.
On the left,
right. Okay.
We have autologous. So it starts with you. We collect your cells, we expand your cells, and then we infuse them. And then on the right-hand side here, allogeneic, you get donors, and usually they put, 5 to 6 donors all in one batch in a big.
So they mix up.
They mix them. Wow. They mix them in a big.
But are they trying to choose people who are close to you genetically, like your kids or anything? No. Wow.
I don't know. They seem to think young Swedish boys are good.
Really?
They go through screening, but there are a lot of complications with this. They don't screen for viruses that they don't know about, or not all the viruses. So how do you know that you're not actually introducing a virus, right, wow, from someone else? There's, there are a lot of question marks. There are a lot of question marks. I personally would never do it. I would never say never. I guess if there's some kind of genetic mutation that I have which requires someone else's cells, I guess fine. But other than that, there's really no reason for me. They put everyone's cells into a big bioreactor. It's just the cheaper option, right?
Yeah.
And your cells, the cells that you have reimplanted, which are not your own, they're allogeneic, they're somebody else's, they're eliminated after 48 hours. So that image I showed you before is not possible unless you take immunosuppressants. And why would you do that?
Yeah.
It's the same concept.
Which is what people do when they get a transplant, organ transplant,
that's
whatever.
Exactly what happens. It's the exact same concept, because each one of your cells has an identity tag on it. And if it's not you, then you will create an immune response reaction.
Sure. Yeah, which tries to eliminate anything that's not you, right?
In my mind, it's like two opposing concepts, right? On the one hand, there's a paracrine effect, but on the other hand, you're actually inducing inflammation.
Right. Cool.
Yeah.
So what else can you show us on your...
Well, we were talking about the totipotent, pluripotent, and multipotent cells. So this is the image I was referring to. So we sit here, and this is the band that you want to stay in. The cells, when they've matured to this band, the multipotent, they don't go backwards. Here, this is the most dangerous. This is when you're a couple hours old. Here, these are the embryonic stem cells. Again, that question mark of cancer kind of resides in these two layers. you don't want to be there, because again, this is a new approach, right? Stem cell therapy, or cell therapies in general, like CAR Ts as well. These are new technologies. We're getting into that personalized medicine space. We're getting into that like very targeted space, right?
Yeah.
I wouldn't mess with the top, the top two categories. You want it to be multipotent, controlled.
So are the top two categories, is that how cancer happens? Is it a malfunction of the stem cell kind of
they're doing their job, because these stem cells, right, they need to replenish their population. And what they do is grow. They do that, right? They divide and grow. They have a population number in your body, right? They're like, because they're the progenitors. And then when you go past that, here we go. When you go above that, let's say, the self-renewal, just don't know what can happen. I mean, there have been some very sad cases. There's this one paper which a lot of people talk about, that they did stem cell therapy, but again, allogeneic, to a little boy. I can't even talk about it. I'm very sensitive to these type of things. I don't remember what disease type he had, but probably a neurodegenerative. And you don't really have a lot of immune cells, right, in your brain and in your spine, let's say.
You don't?
Not many.
Right.
No, you do have, but not many. And they kind of reside there. Your blood-brain barrier. I mean, so stem cell therapy, even your own, they don't usually pass through the blood-brain barrier. Although when you have a neurodegenerative disease, it's much easier for the cell to cross over. Exosomes can, but it's anyways much more difficult. So in those instances, in that disease, neurodegenerative diseases, it actually has shown a lot of promise.
Right. That's good.
So anyways, so in this study, they found that young boy that they treated with allogeneic stem cells actually had a tumor growing on his spine.
Oh, wow.
And they found many different cell types. Many different cells, tumors from many different people.
Right.
Yeah, exactly. Because that's what they do. It's kind of like a mishmash of
I mean, it seems to make just
cells.
natural common sense that if you're to use your own body, which your body's used to and recognizes, would seem to naturally be more aligned. I mean, on the scientists, but yeah.
I mean, but everybody knows that if you have some kind of organ transplant,
then
it gets rejected. It's the same
for the
exact
rest
thing.
of their life to take the
immunosuppressants. But I
drugs.
mean, you're not really going to take immunosuppressants for osteoarthritis, are you?
Sure, no.
And it's just better to stay away from those. If you can, you stay away from those types of drugs.
I mean,
you don't want to suppress your immune system, right?
I didn't realize you could get stem cells just from like behind your ear. I thought it was only specific circumstances like the umbilical cord, or I've also heard from your baby teeth. Is that not true?
Yeah, that's true.
So you can say, so is it baby or any teeth?
I'm pretty sure it's any teeth.
Right. So my daughter's has lost a lot of her teeth. We've saved them. Is that good? Can that be then used for her later at some point?
I don't think you can. I don't think later, because you want a live cell, right?
Oh, so
you'd have to do it immediately.
Okay, what, like freeze it? Oh,
well, first of all, you have to harvest it.
Okay.
Right, and then they'll freeze it down. Yeah. I mean, we don't do it because again, it's a specific protocol. But you can do it. There are places that do it.
Interesting. Cool. So what are the sort of moonshot ideas that could come out of this, or what that you hope could be where could this go in terms of
my goodness,
human healing, human
it really can
possibility.
Go anywhere. And this is the sort of thing that people are doing right now. Like it all starts from harvesting your stem cells, and then there's obviously genetic manipulation, right? You can edit the gene of your stem cells, like what you do with fibroblasts, iPSCs. I mean, really Bob's your uncle. But again, it's early-ish stages. We have these CAR T therapies. But then you have to go through, again, you need to go through regulatory approvals. You need to have clinical trials conducted and prove that this approach is safe.
So do you always have to do that with humans, or does it start with animals first and then move to...
Oh, yes, animal. Well, you need to show your in vitro data. So you have your cell models. You see how things kind of
react.
Yes, exactly. You're looking at the life cycle of that cell with your whatever manipulated, for example, if it's gene therapy, and then you move on to the animal model, then you present that to the regulators. If they're happy with your results, then you move into your phase 1, and then you
that's
grow
like kind of
your
volunteer human trials. Is that volunteer human trials?
So, yes, volunteer, but when you do clinical trials, there's also, you have to think about what indication you're going for. So what disease are you trying to treat? What are you looking at? And then you get into like a specific line of therapy. So you have first-line therapy, second-line therapy, third-line therapy, fourth-line therapy. So there's like the standard, like the NICE guidelines, there's a standard of... there's a standard approach, a standard drug that has been chosen for that specific indication. If the patient doesn't respond to that indication, then they move on to the next drug that's in line. So you didn't respond well to that, then you go to your second line. You didn't respond well to that, you go to your fourth line. So that's common, for example, in cancer, because we have a lot of drugs, oncology drugs in the space. And they could say, like, for your phase 1, you're allowed to treat patients that are in the fourth line therapy. So they've already tried 3 drugs. They're not responding well.
They were resistant to others.
Yeah,
drugs,
and I mean, at that point, they're hoping maybe that's a miracle that something will
help. Then they may as well
there.
Basically try something new. Yeah. So that's how they justify some new treatment.
Yeah,
I see.
Justify it. I mean, and if it was me, right, and I mean, I was in that position with my mother, what can happen any worse than the death sentence that she was given?
You've got nothing
right?
to lose at that point. Yeah,
exactly.
Yeah. So you're starting.
So you're not just given whatever patients to make your guinea pigs.
Sure.
But yeah. You have to, because you need to show efficacy and you need to show safety.
Right.
And depending on the indication, are you going for orphan drug? Orphan drug is like rare diseases. So it takes about $1 billion to take a drug from bench to market.
Wow.
And I don't know what the stats are now,
but that sounds a lot.
Yeah, it's a lot. It's a lot.
What's involved, I mean?
You can't imagine. And it's something that people are not aware of. And a lot of that money that pharma biotech has invested literally goes down the drain in like a phase 3 clinical trial. So you've already spent a lot of money up until that point. And then suddenly, when you're looking at a bigger N, the population size of your trials, so it's very different when you're treating 8 people to 2,000 people. The data, it can go completely the opposite direction.
Yeah.
So
you have a larger sample set. Yeah.
Huge difference. And then suddenly you have to scrap your clinical trial. So that's bye. Everything.
So the stuff you're doing has gone through.
Well, we've treated 16,000 patients.
Right.
You usually don't get to that number throughout the whole process of the clinical trial.
Right. So that's equivalent of having gone through that because another country's done it. And then that's in a variety of applications for various different diseases then.
Yeah.
Very cool. 15
indications we have in Japan.
Very cool. Well, would you like to give a little summary of your clinic and where it is and how to find you and when you're open and when can people use your
when do people use
treatment?
Us? Well,
yeah. When can we come and fix ourselves and boost ourselves? And
that's up to the MHRA, but their responses are very quick. So we're hoping beginning of the year we'll be able to actually treat. Of course, we can harvest your stem cells.
Already? Okay, so harvesting's allowed.
Harvesting, we have our CQC so we have various regulators. It's not just the MHRA. We have CQC. We went through that. They were also amazing, really amazing. The person that we were dealing with in the end, she had a PhD in the space. I spoke to her personally. We ran her through everything that we do. And we couldn't feel more supported. They were fantastic, actually. I have to shout out to them.
Okay.
And then the MHRA and the HTA, Human Tissue Authority, we don't technically need it because we have our operating theaters adjacent to the lab. And they told us ourselves, if you build it.
So where are you now with the progress towards actually offering the treatments to the public, and where can they find you? Share your socials or whatever, or your website.
Well, we want to be very conservative. We're scientists, and this is a scientist-led facility.
Right.
And we're very prudent. We don't want to promise too much. We want our results to speak for themselves. We are born and bred scientists. We don't believe in too much marketing, and you just have to be careful and mindful. But again, we have the data to back it.
Mm-hmm. I'm sure you have investors that are looking for a return.
We're self-funded.
Oh, you're self-funded?
Yeah,
okay.
We have people that have approached us a lot. But I think it's important also for us, we need graft. We need to experience it ourselves. I mean, everyone, my whole team is really vested. And we've all shed blood and tears. Not just me, not just Christina, our CSO, and my partner. We have all shed blood, sweat, and tears. We laugh hard, we cry hard.
What's been the most challenging part of it all? Is it the regulations or...
the build.
So you were saying the most challenging part has been the actual physical build out.
The physical build. Again, as I said, we have 2 meters of technology. When I took over the space, when I leased out the space, so by the way, we're in Mayfair. So also that
that must be really tough to do the construction there, because it's not the easiest place with access.
No, it really, I can't tell you how many times we almost went out of business before we even started.
My God.
Liquid nitrogen, because we have to bring liquid nitrogen on site.
Right.
And the regulations generally have changed, and people don't like delivering to London because liquid nitrogen, it's a hazard. It's dangerous handling the liquid nitrogen.
Explosive? No.
No.
It just freezes
quite the
things.
opposite. It displaces oxygen. You suffocate from it.
Oh.
Yeah.
Okay.
So you have, but we have all the necessary protections in place.
It's funny how the toughest thing is the construction rather than the actual business itself.
Yeah.
Yeah.
We've got it locked down, the science part, right? We've already gone through all of that stuff.
So in
our
Japan,
background, right? Well, no,
so
but us as well, right? Because it's cell culturing, of course. Yeah, we've done all of that in
the
King's.
UK.
Yeah. Our academic side.
Right.
So we have that experience, but building such a high-tech facility. We're talking about sensors behind the walls, in the ceilings. All these things are fickle, right?
I'm sure you lose so many subcontractors with different companies with specialists in different areas.
We actually have Norwood was our contractor. They're specialists in clean room design and build. They have been phenomenal. They've been fantastic, really. They build the grade B panels. So they specifically build those panels for us in their manufacturing facility.
Right.
Everything is specialist. Every single thing, especially in GMP, it's even more specialist than just a normal, let's say, clean room, because again, you have different grades. Just so you understand the difference, and every ingredient that we use and anything we use is way more expensive.
So your supplier list is very filtered and specific.
And we have to audit our suppliers as well. Just an indication.
You have to order them.
Oh, yes, we do. Mm-hmm.
What?
Us
go
ourselves.
to their facilities and make sure they're doing what they say.
Yeah, we have to, and questionnaires, and we have to vet them. And we have to show that to the MHRA, to the regulator.
Quite a big
I mean, it's
team to help you with all that?
We don't have such a big team. It's better when you're lean.
Yeah.
I mean, I think that every single person on our team can build a lab.
Really?
Now they can literally are manufacturing scientists.
How many are you?
Altogether. So we have some people that are not in the UK. I've brought on my mentor. Dr. Nasos Alevizopoulos has been my mentor, and we've worked together for over 20 years now. Oh, wow. And I don't move without him because I know he's a stifler. He's like, everything needs to be perfect. He's very cautious, and you need someone that's like that. And he knows everything about everything. It's crazy what we were talking about before. He has an MBA and a PhD from Lausanne. He has written 4 INDs, so dossiers for the FDA. I mean, that is quite an undertaking. And now he's doing ours together with us. But he knows from the molecular side, like the research side, to how you validate that even in vivo, and what type of tests are required and what is expected. He is meticulous. So you need someone like that on your team.
So has he run labs previously then to know that much detail, or like in Japan or anything?
He did work at Novartis. I don't know that he's actually worked in labs per se. I mean, we've all worked in labs, right? Because our PhDs are in labs.
Like the wrong jargon, but I mean, like
he's got
operation.
He has a lot of experience.
Right.
I mean, I don't know many people. I think he's the only person that I know that has written 4 INDs, you know, in the oncology space. And I mean, this was definitely a challenge, and he was skeptical at the beginning, and then he looked at the data and he was like, oh my God, this can actually be
revolutionary.
Yeah. And now I believe he's drinking the Kool-Aid.
Oh, what is he?
No, he's... we haven't taken a sample from him yet, but you see, the reason why we've taken our own biopsies before everything is because we need to do our validation run. So we need to do our whole process in the lab to show the numbers, and, you know, if the samples are sterile. So we do actually, circling back to what we started saying before, twice in our process. So we have our QC, our quality control, right before we release the drug. You need to have a QC done. Like, is there endotoxin in the sample? Is there mycoplasma in the sample? Is it sterile? So we do all these tests and say, okay, this is ready to go. So we compound it. We go through that process, close it up in a vial. It's ready to be injected. So we do that even before our release tests occurs. We test these samples sort of at the beginning towards the middle, like before we bank the sample.
Right. So, and then once it's banked, then it can be frozen for years, or?
Yeah.
Right. That's cool.
Yeah.
Okay, so do you want to share your website, or how do we find you? How can people that want the first humans that want to get involved from January, or like when are you offering treatments, or do you expect to?
We expect,
because you can't say it.
No, exactly. But we have had a lot of interest, and we're building our waiting list, because a lot of people want to be the first. I understand that. Yeah. Sorry, guys, I'm going to be the first.
Right.
But, you know, because this is so specialized and it's personal, it takes a lot of people for one person's drug. Right. So our capacity, and we want to keep, we want to be controlled. Our capacity is not...
too extensive,
yeah, it's not extensive.
Well,
exactly.
I'm just imagining you going through your airlocks and taking off all your, putting on your gloves in the right order, whatever, just for that. I mean,
and then the cleaning and
all that just to keep it
exactly. So so that waiting list is growing.
It's growing. Yeah. Well, and in the new year, you expect 2026. I mean, we can take
biopsies.
Yeah,
and we can bank the cells. But so then they'll be ready to go.
Right.
Ish. Again, we need a couple steps before we actually reimplant. But hopefully in January we'll be.
And how can people find you?
We'll be there.
What's your
What's our address?
Yeah. Well, you're in Mayfair.
We're in Mayfair, 1 Brook Street. So we have a fantastic address.
1 Brook
We
Street.
we wanna be easy to find, and we're in a beautiful new building, which was also one of my prerequisites. I'm not a big fan of Harley Street. I think
Of
it's
the
kind
old,
of, I mean,
dingy rooms. Yeah.
I don't like going there myself as a patient, and when you have such a high-tech
Creaky
lab,
wooden floors.
exactly.
And,
Like I can't have mold
in
in the walls. I can't have a potential like a pipe burst because the pipes are like 100 years
Victorian,
old.
whatever.
Exactly. So our building is brand spanking new. I mean, literally, we built it out from a concrete shell.
Oh, nice. So then you got to design it properly, everything from the ground up.
Exactly. No,
Okay.
I mean, I was there, on my knees with chalk.
Oh, really?
Yeah.
I'd love to have seen that.
Measuring out the spaces. So,
Cool.
we've really, we put our heart into this.
Well, we'll add a link to your website and your socials down below for everyone to get in contact. And thanks for sharing your incredible story, and good luck with being the first stem cell clinic or autologous stem cell clinic in the Western world.
Stem cell center.
Oh, center.
Yes,
okay,
please.
you're not
because it's a whole facility.
Okay,
it's not
it
just
is.
a clinic.
End to end.
Yeah,
very cool.
end. Yeah, exactly. Farm to table under one roof. Awesome.
Thanks a lot, Timi.
Thank you very much for having me.
Yeah.
It was so nice to chat with you.
Yeah.
This is my first podcast.
Wow.
So there we go.
Well, the first of many, I'm sure.
Yeah, well, let's see. Hopefully, we're very we're interested in educating people about this. I think people need to know. I mean, if you don't come to us, that's fine. As long as you have the tools to identify, the charlatans out there, because it's dangerous.
Yeah.
It's dangerous. You need to know.
Well, you sound like you're going through all the right processes to keep it safe. So
it was for my mother and someone else's mother, and just the way I want someone to treat my family.
That's how you treat others.
I would expect that for anybody else.
Perfect. Okay. Thanks
Perfect.
a lot.
Thank you so
Cheers.
much. Have a good day. Bye.